Okay, everyone, I'm going to be picking up where I left off in that first nervous system layout lecture, the very fundamentals of the nervous system. And I've decided I'm going to hold off a little bit about going back to the cell membrane, perhaps even until we're doing neurophysiology the way that I want to discuss it. So let's go back to what I had kind of finished up with in the part 1 video, and that was discussing the different classes of neurons. And I gave you some examples here of the basic neuron types, also those special definitions of clusters of cell bodies in the CNS versus the PNS, and also those bundles of axons that necessarily follow from those cell bodies, and that their names differ in the CNS from the PNS. So let's move on to the next page here, and we will go to glial cells. Glial cells are not neurons. And again, they have a couple of names in the CNS. They are called glial cells, but if you move into the periphery, the peripheral nervous system, remember? Okay, they are called satellite cells, and I think that kind of is easy to remember because satellites are out there. Anyway, once it was thought, the only thing they did, which actually is not a small thing, was to provide physical and functional support. The word glia comes from a word that means glue, providing a physical matrix in which neurons could exist and function. It was found later that they play a role in the biochemical environment, that they actually take place in the exchange of certain chemicals. Now, it's actually believed that they are also active in the modulation of certain neuronal activity. So they are now considered to be much more than a matrix that just is supporting everything else in the system. There are different classes of glial cells, just as there were of neurons. The first is the astroglia. I've heard people say astroglia. Doesn't matter. It's the same thing as astrocyte, cyte meaning cell. And they are the largest of the glial cells, and they cover blood vessels, okay? So they can cover around blood vessels, and they also make contact with neuronal cell bodies. So you're like, whoa, this could be important. And they exchange chemicals. As you see above, when I said there's exchange of chemical in that biochemical environment, they do appear to exchange chemicals with adjacent neurons, whatever neurons are next to them. And I want to mention a very special, cool form of glia. I love these glia. It's called radial glia, and these are a specialized type of astrocyte. And their first job is to guide the migration of neurons during embryonic development. This is critical. If these neurons don't get to the right areas, we do not have an appropriate nervous system, if one at all. So we want these neurons, these radial glia and neurons, to get to the right place. So radial glia, very important, little guides. I almost think of them in some ways of being like a lattice for flowers or plants to grow up and get to a certain place. Later, though, once they guide their neuron to its proper place, they don't just kick off and go, oh, my time is done, or else I just may as well die, my job is done. No, they transform into what we call a supporting astrocyte. And here you can see that, whoops, sorry, backspace. Here you can see these are real images of astrocytes, real astrocytes. So Very cool cells, very important. Radioglia among my favorite. So here comes microglia. Microglia, if you must. Right away you see micro, that should tell you immediately that they're smaller than the astrocytes or astroglia. Their primary job appears to be removing waste materials. And I'm telling you, this is also an extremely important job. I just know, like, if you let waste accumulate, it not only is unpleasant, it gets to the point where it taints the environment around that waste. And it can turn things into toxic materials. Just think of it in a way where if your crew who comes to take off your garbage cans and put all that stuff somewhere and take it away, if they never came. So they're considered scavengers because they just kind of go around and they find this waste material here and they pick up, and waste material there and they pick up. So this again is an image of a slide of these microglia. And again, they're smaller, should not be surprising, than astrocytes. I put this little extra diagram down here for those of you who are very excited about the cellular bio aspect of this, and you can kind of get a sense of how they operate, how they develop and operate. Not going to be necessary for questions and exams. So. Now let's talk about oligodendrocytes. They actually form myelin sheaths around the axons of some of the neurons in the central nervous system. Oligodendrocytes. And oligo kind of comes from the rule by one or just a couple, like, instead of rule by many. So these guys are really cool because what they do is they reach out. Here is the cell body of it, and then their processes proceed from that cell body and go and wrap around different segments of those axons. And so one guy looks a little bit like a squid or octopus in a way, and can reach out with tentacles and surround different areas on the same axon or on different neighboring axons. Okay, and they're important. Myelin is a lipid, and it is very important for insulating these axons. Remember, we have electrical conduction, and it also play a role in increasing the speed of transmission of neural messages. When we get to neurophysiology, we will be talking about how these particular cells contribute to that insulation and to conduction of neural messages. Now, if we move into the PNS, we have these cells, the same thing that form myelin sheaths, a glial cell, called Schwann cells. This is in the PNS. And in this case, we have one cell not able to reach out and surround multiple segments of multiple axons, but one cell results in putting out one myelin segment. And these are important for guiding axonal regeneration. If you cut an axon, they can be useful in guiding that to the right way again, if the actual cell itself is healthy enough to do that. Now, just to look here and compare to what we saw with the oligodendrocyte, here is the cell body. And what we have is this cell body giving itself up and going, okay, I'm going to surround and surround and surround and surround and give my whole self up to surround just this one segment of the axon, just one region. I kind of look at it as like, whoops, sorry again, paper towels, a roll of paper towels. And this would kind of right here be the cardboard roll inside. And then let's say we have this little cell that gives itself up to wrap the paper towels around and around and around this one segment, this one cardboard roll. Okay. So that was brief in this segment. I wanted to kind of break it up a little bit. I had thought about doing something a little extra with it, and then I thought twice about it and decided to move it later into the neurophysiology part. So this is just a baby lecture. If possible, you should do all 3 of the sections at once that I've put up for you. But if you don't, they're here for your convenience, for your flexible schedule, and hopefully that helps. We will pick up with the development of the central nervous system. Really cool.